So, transport through the cell membranes, the phospholipid bilayer is actually a really good barrier around the cells. It's especially a good barrier to water-soluble molecules, polar molecules. But we really need some stuff to cross to be able to enter and leave that cell. There are 4 basic mechanisms for the transport through the cell membrane. There's diffusion, and facilitated diffusion is a type of diffusion. We have osmosis, and then we have active transport and bulk transport. The first 2 will not require energy. They will be passive processes. Types 3 and 4 will be active processes. They will require energy on behalf of the cell. Active transport usually requires direct spending of ATP. Bulk transport usually requires movement of the cytoskeleton and a whole bunch of other stuff to happen. So here's diffusion. Diffusion in liquids is a good example, a dispersal of dye in water. So diffusion is the net movement of molecules or ions from a region of high concentration to a region of low concentration. And this idea of high to low is going to follow you for the rest of AS Bio, for the rest of your life. High to low. Everything moves from an area of high to an area of low. That will always be true. Your job is to phrase your statements in a way that is conducive to what is the high area, what is the low area, and where is the direction of movement. From where to where, by what means. That's your job. That's your job in a nutshell. So molecules during diffusion are going to move down a concentration gradient. Molecules all have kinetic energy, which makes them move about randomly. So as a result of diffusion, molecules will reach an equilibrium, where they're evenly spread out. Equilibrium is where there is no net movement of molecules to either side of a membrane. No net movement. That means that for every particle that moves to one side, there should be some same number of particles that moves to the other side. So there's no net. Your income is the same as your expenditures. You have no net profit. So passive diffusion means that there is no energy used when molecules move. They have a natural kinetic energy that is always just going to move them. So these blue particles are going to naturally, passively diffuse through the red liquid. Just passively. Until they are reaching an equilibrium where there is no net movement or no change in concentration of the blue dye throughout the entirety of the red liquid. So in the diffusion of bromine gas, we're going to have a high concentration of bromine at the bottom of the glass vial and diffuse to an area of low concentration of bromine at the top of the glass vial. It will move by diffusion. So, when we talk about diffusion through a membrane, we have to understand that membranes are semi-permeable. They can only allow for the diffusion of certain things. Whether those things are specifically non-polar stuff, molecules of a certain size cannot pass, molecules of a certain identity, molecules of a certain charge, they are not able to pass. So this cell membrane, let's say that these molecules are both non-polar, but we have molecules of a certain size are only able to pass. Only the red molecules are able to pass. Eventually diffusion will occur until we have reached equilibrium of the red molecules. The red molecules are in equilibrium, where there is no net movement back and forth of the red molecules. The blue molecules are still remaining behind. They cannot diffuse. They have no means by which to move from their area of high concentration to low concentration. Now, one might think that, well, shouldn't there be the same number of particles on both sides? No, because diffusion only matters to one particle at a time. It only matters to one solute at a time. So if you're talking about the diffusion of sodium, you're only dealing with sodium. If you're talking about the diffusion of potassium, you're only dealing with potassium. If you're talking about the diffusion of methane, you're only dealing with methane. If you're talking about the diffusion of cows, you're only dealing with cows. You're not dealing with chickens, not pigs, not geese. Only cows. So, assuming that molecules are going to be small, hydrophobic, they can diffuse in and out of cells freely. So if they are small and hydrophobic, that means they will spread evenly. Molecules move constantly and randomly. True. Over time, they will distribute themselves evenly. True. Small hydrophobic molecules can diffuse in and out of cells. So, I would amend this statement. Over time, they will distribute themselves evenly if they can. And small hydrophobic molecules can. So these small hydrophobic molecules can diffuse out. These small hydrophobic molecules can diffuse in. So they will. Freely. So, what determines the rate of diffusion? There are 4 factors that determine the rate of diffusion. The first is the steepness of the concentration gradient. How high is your high concentration, and how low is your low concentration? If you have a great difference between your two sides, if you have a very high concentration gradient and a very high concentration and a very low concentration, you have a steep concentration gradient. If you have an even higher concentration, but then you only have a concentration that is low to there, that's all you got. That's not a very steep concentration gradient. This will be a faster rate of diffusion than this, even though this is a higher concentration to start. It's the concentration gradient, the difference between my high and my low. The high and my low. It's always the difference, the steepness of my slope. Temperature. Because molecules are diffusing thanks to kinetic energy, higher temperatures give molecules kinetic energy. So if you give molecules more kinetic energy, they will diffuse faster. Greater kinetic energy yields greater diffusion rate. So higher temperature, higher rate of diffusion. Period, end of story. Surface area. So if I have a membrane, if I am not allowed to diffuse through here, and I can only allow the crossing of stuff through this space, I will be able to pass 2 at a time through here. If my surface area is only that wide, only 1 at a time may pass. My rate of diffusion will be faster through the larger surface area. Larger surface area gives you greater diffusion rate simply because more molecules can diffuse at the same time. So, more molecules or ions can cross the membrane at any given moment. That is how surface area increases the rate of diffusion. If there is no surface area, if you are just putting dye into water, surface area is effectively infinite and you have no problem there. That is not a factor if you're just dropping stuff into a bowl of water. But if you are diffusing things through a membrane, that becomes a factor. Now, the type of molecule or ion that is diffusing. Remember that we are dealing with a phospholipid bilayer, which has a non-polar fatty acid core. This non-polar barrier will